Why don't we get started with the introduction and then we can go with the speakers. So again, for all of you online, I hope you can hear me. Susie Fitzpatrick, one of the organizers of this meeting, and thank you for coming to webinar 2 of Trust Your Gut, Establishing Confidence in Gastrointestinal Models. Next slide. So the background is, we know the first question that you ask when you're looking at a orally administered compound is how much bioavailable is it? How much is really getting into the gut? So we want to look at how different models can provide insight into this to gut absorption. We want to emphasize that you should ask the question first, what are your questions, and then develop your tools in order to answer those questions in hand, regulator and researchers doing these new models, which will help us, I think, really advance working together to get them into regulatory use. So do you need, for example, a microbiome, peristalsis? Are you looking at allergenicity testing, etc.? So this webinar is laying the foundation for these upcoming workshops. I hope that you can attend these. The next one on allergenicity, if we're working and not on furlough, and then come in person to NIH to really hear some of the different models and talk to us about how we can establish confidence in them. So next slide. So this is the organizing committee. It was really a great cross-sectional group of people from several different federal agencies. We'd also like to thank the T4 Transatlantic Think Tank out of Hopkins for their support, and thank you for ICFAM and NICEDM for helping us put this together, especially Amber, who did a great job of herding us all together and getting it done. Next slide. So again, save the date. These two are, allergenicity is October 6th. The workshop itself is in person at NIH or virtual. I'll encourage all of you to come in person. It would be great to see everybody. And I think that's going to really help the dialogue. And that's October 11th and 12th. And again, we're looking to see if we're working then. So keep track of what's going on to know if these things are still working. Next slide. So the two speakers today, we're lucky to have our European colleagues come. George Cass from EFSA is talking about the gut as a target organ and barrier challenges. Jose from PARC, used to be at EFSA, now at PARC, new tools for mechanistic understanding. And then we have time for some questions. Send your questions either into the chat or raise your hand. Next slide. So rename yourself in the window. You can see the instructions here. The toolbars are accessible at the bottom of this webinar, so you know where the chat is. And next slide. Speakers will answer clarifying questions at the end of their talk if we have the time. And as I said, either raise your hand or put it in the chat. Next slide. And this is being recorded for those of you that want to either listen to it again or for your colleagues that can't make it today, if you could let them know that the recordings will be available at this website here. Next slide. So with that, it's my pleasure to introduce George Cass. George was trained as a biochemist. He received his PhD in biochemical toxicology from the Karolinski Institute. Then he worked at the Swiss Federal Institute of Technology in Zurich, and then returned to the Karolinski Institute. He's been a very great colleague of FDA, where he works at the European Food Safety Administration since 2009, the lead expert on toxicology. And again, he's registered in the UK Register of Toxicologists and in Eurotox. And again, a really great colleague. And with that, I'll introduce George and take it away.
Right. Thank you very much for the kind introduction, Susie, and good morning, everyone. I hope you can hear me. That should work. I will start sharing the screen, and while doing so, I will take the opportunity to thank you for inviting me to participate to this seminar and to give this presentation. Really grateful for that. what I want to do is give you sort of an overview where we are in Europe in terms of chemical risk assessment, primarily focusing on the regulatory side, on the decision-making side. The issues we have dealing with a lot of legislations, a lot of procedures, much based on traditional tech approaches, traditional in vivo approaches, and how we see the future in trying to move into what we would call the next generation risk assessment, and particularly using in vitro models, in silico models, to help us progress in the way we do the risk assessment. Yes, it works. So first, a disclaimer. So what I'm sharing here with you, of course, my views, my thoughts, my opinions that are not necessarily those of my organization. And I would like to start setting the sort of a regulatory scene as it is in Europe. But of course, this is quite applicable also more widely, more globally, because legislations are there for chemical risk assessment to protect the consumer, to protect the public in general throughout the globe. What you can see here, this is what the European situation, and of course, I don't know whether the mouse works here, but on the top left, you have the legislation dealing with food safety, the primary legislation for food safety, but we also have legislation for industrial chemicals, and you have on the right-hand side, so this is the REACH legislation, which is, I believe, sort of the analog over in Europe of the Toxic Substance Control Act you have in the US. And then we also have other sectors that are legislated. Just to give you some examples, of the toy directive, but also one I want to highlight to you is the ones on cosmetic products, which of course has a number of implications in terms of the use of animals and animal testing. So to sum it up at the European level, of course, when dealing with environmental pollutants, there is no formal testing as such. However, in all the other sectors dealing with pharmaceuticals, food additives, plant protection products, biocides, etc., extensive testing. So all the chemicals have to go through an extensive battery of testing to be able to evaluate it and then to be put on the market. When it comes to industrial and consumer chemicals, this ranges from limited testing to extensive testing. This is very much dictated by the amount being put on the market or being produced or being imported. This is for the EU. And then we have the sort of the slightly different situation where the cosmetics, where in fact since 2013 it is prohibited to use animal data and then to generate new animal data for the testing, for the risk assessment of cosmetics. So coming back to the food and feed side, so EFSA was established back in 2002. It was very much the consequence that the situation we had in Europe, taking us back into the 1990s, even the early 1990s, where we had a number of crises, primarily the so-called mad cow's disease, so the spongiform encephalitis in bovine. We also had a dioxin crisis in Europe, and so the confidence in the EU food supply was very much eroded in Europe, and it was clear to have something at the European level, also to ensure that the consumer is very well protected, and also to improve the whole food safety system within Europe. And what this law has also done is the separation of risk assessment from the risk management. What we are doing as an organization, we provide independent scientific advice and support to the risk managers and the policy makers in Europe. We communicate this information. So our role is to provide timely and independent risk communication. And of course, and also one of the reasons I'm sitting here and sharing the information with you. We promote scientific cooperation. We do that at the European level, at the national level in Europe, but also very much at the global level with all our partners in different parts of the world. To bring back, to focus a bit more on what we're doing at the European level, and this on the left-hand side, you see also the EFSA atom, very much representing the different sectors where we're active. So we're talking here about ranging from farm to fork. We're talking about plant health, over to plant protection products, the GMO aspects fall in our remit. We deal with animal feed as well as animal health and welfare. We deal with hazards, whether they're biological, so talking about zoonosis here, or as chemicals in the form of contaminants. In other words, about chemicals that are not supposed to be in the food but end up in the food chain, or talking about, or also talking about chemicals that intentionally added to the food chain. For example, food additives. We also deal with chemicals that are used to protect foods in the form of food packaging. And finally, we also deal with the wider nutrition aspects for the human population. In terms of types of chemicals we're dealing with, so we separate, as already said, the contaminants, where there's no commercial producer, from the regulated products where we have very clear applicants. And much of my talk will be focusing more on the regulated products, where we also deal with new chemicals as well as chemicals already on the market. So part of our remit is also to reassess chemicals that are already on the market, reassess for their safety, to see if and give advice to the risk managers to reauthorize the chemicals on the market. It is, for example, very typical for plant protection products in the EU. So to set the EFSA scene, what we are dealing with, I've mentioned already, regulation that at the wider EU level, but also within our own sector, our own activities, we have a lot of regulations, something over 30 regulations alone dealing with food and feed safety. And what you find there, in some of the legislation, you will find that the type of data that are being requested to be sent to EFSA as an organization are stipulated in the legislation, but often this is not the case, and the type of data requested is very much left to EFSA as an organization, and this is done in the form of guidance documents that we are developing for all the different sectors, whether it's from food contact materials, whether it's for additives, whether it's plant protection products, etc. Not plant protection products, because that is already in the legislation. For food enzymes, for example, we developed these guidance documents where we very much let's say share with the consumer, but also for the applicants, what side of data is required. If we look at the European level and the type of data we get and the type of data we request, this is very much driven by the exposure side. This is, let's say, a more generalized thing. There are also exceptions here, but if you look at from a high-level perspective, you see that if the exposure is low, if there's a low use of a chemical, the requirements are less than what you require for a high-use chemicals where you're expecting a high exposure. And the typical type of data that is being required really at the low level of exposure will always be the genotoxicity. Certainly in the food sector, no substance can be put on the market if it harbors a genotoxic potential. And of course, also need to know about the ADME properties. And as with the requirements increase, we go from having a request for subchronic toxicity testing, getting full ANME information, up to having the full package of the CARC studies, the chronic toxicities, the reproductive developmental toxicity studies, and sometimes also specific or specialized studies. So if we look at the nature of the data and how they're being generated, you'll see that when it comes to the ADME side, primarily, so what we call the biological studies, they all come from in vivo. They have to follow OECD test guidelines, and they have to meet GLP criteria to generate the typical TKE parameters. For the chronic side, again, these are produced from in vivo experimentation. They have to follow OECD test guidelines, GLP criteria, and this study would provide us with the traditional tox parameters. There's a bit of room for in vitro studies currently. This is restricted to the genotoxicity studies, certainly in the first phase, and also some metabolism studies. For example, there's a need for doing comparative metabolism for plant protection products, and this is done in vitro to a large extent. Again, they have to follow OECD test guidelines and GLP criteria. So what I want to say here is that currently, and here we're talking about 2023 in Europe, certainly in the food sector, food and feed safety sector, the traditional chemical risk assessment relies mainly on animal bioassays, and the same is also applicable for the data required for classification, for example, under REACH, for industrial chemicals. So this, if we take it, let's say, from a traditional point of view, has been the way we operate it. Of course, things are changing. And what is, for example, changing is also what we're dealing with. So we are dealing now with new challenges. We're dealing with new threats. This can be for the human side. It can be for the environmental side, alone for the mental side, the issue of multiple stressors and the impact on bees is, let's say, as a perception new, and we have to deal with it. But also for the human side, for the consumer, we have novel foods coming onto the markets. We're dealing with nanomaterials. We're expecting nanopesticides, for example, to sooner or later being at least doses being sent to EFSA in the future. And we're dealing with chemical mixtures, not that they're new, but we have to address them. The public is much more conscious about this. Consumers more conscious about this, and we're also aware that we really need to look at what is the impact of being exposed to mixtures of chemicals rather than single compounds on which we base the traditional risk assessment. So things that landscape is changing, it's changing rapidly, but also at the policy level, we have a lot of changes occurring in Europe. For example, we have the Green Deal that was announced a few years ago, and this was followed up by the EU Chemical Strategy for Sustainability. And here already in that document, there's a clear mention that for the safety testing and the chemical risk assessment, we need to reduce the dependency on animal testing, and we also need to improve the quality, the efficiency, and the speed of chemical hazard and risk assessment. So the Commission will support initiatives and for innovation to advance the tools for doing our assessments. This is also reflected in the EFSA strategy, the 2027 strategy, where we also make very clear statement that a key action will be to develop and integrate new approach methodologies, NAMs, and omics for regulatory risk assessment. And as we have collaboration as one of the key pillars in our activities, it's clear that we work together with a number of organizations and a number of initiatives at the European level, for example, with some preview projects such as PARC and the ASPIS Consortium, but also at the international level, we're engaged in APCRA. We are engaged in the Global Coalition for Regulatory Science Research. We work with OECD etc. So we are very much engaged in developing new approaches for chemical risk assessment. How are we going to implement these changes? Well, we see it a bit as a sort of an evolution rather than a revolution. And what we want to see more is a sort of a more holistic approach, really bringing closer the exposure side. Oops, sorry, I'm going to go back. The exposure side, getting more holistic information through exposome, working more with epidemiological data, getting more exposure data from human biomonitoring, but on the hazard side, really moving away from the classical in vivo tox into bringing into the equation what we call the NAM data galaxy, with all those new information that we can generate, and using this to really bring it together into let's say an advanced risk assessment and risk characterization, and hopefully also in the future move to next generation risk assessment. We invested already considerably in this. We have invested a lot into project. We have something like 20 million euros. It's similar equivalent to about $1 million into projects. They've been instrumental in providing us case studies and information to allow us to update or develop new guidance documents where we really integrate NAMs. Often, it's fair to say that the in vivo remains a major part, but we're developing gaps, or we're looking at gaps where NAMs can actually help us to come to a better and faster conclusions. One perhaps to illustrate is the current guidance document that we are developing on read across for food safety assessment. So let's go to the topic of this workshop, these presentations, the absorption, the pharmacokinetics, and why are we very much interested in this? Well, one of the approaches we're using, and that is reflected in some of the guidance documents, so we're talking here about food additives, novel foods, and also nutrient sources, is a tiered approach. And just to illustrate what we've done for the food additives, and this already is 10 years old. So this was published in 2012, and of course, all these guidance documents are up for renewal, and we're looking at updating and improving on them over the coming years. But just to share with you where we are and to see where we can go from this. So having the tiered approach means that we can have an approach where if we have information on absorption, the genotoxicity of the compound, and also the local toxicity of a compound, and we can prove or if the applicants can prove at the level of absorption that the compound does not get into systemic circulation, so it does not or even reach the liver. So there's no bioavailability or limited bioavailability, then we can stop at this first tier, which means that we do not need to go into the traditional repeated dose studies that are normally requested. So if the compound does not cause any GI toxicity, does not give any alerts for genotoxicity, its absorption is negligible, then we can stick at the tier 1, and it's really that we do not need to undertake any higher tiered toxicological studies. So fully in the level, fully aligned with the 3 Rs. We also have an approach for the nano. So how to assess nanoparticles and how to ensure that they are safe. We're also we're looking at absorption very much. And the nano guidance really gives us options that we call exit routes. So to getting out of the nano assessment, and very much of a driver is the ability of nanoparticle to be soluble in the aqueous environment, so to be soluble in the GI tract. And if, of course, the solution is fast, then there is an exit route so that the substance does not need to be studied as a nanoparticle, but as the chemical itself. So this is illustrated here, and below you have the guidance for this. So if the nanoparticle is soluble in water or soluble in the marketed products where it doesn't even reach the consumer as a nanoparticle, and it's rapidly dissolved, then the material can be considered as bioavailable, and the conventional risk assessment can be applied. It also means that if we already have information for the conventional material, then there's no need to get into new studies. It also means that if the particles are not absorbed, but they are large in size, so they're not nanoparticles, so we only have 10% or less than 10% of the particle is smaller than 500 nanometer, then we assume that the uptake will be negligible, and again the risk assessment can stop there at that level. If of course it's not, if there's nanoparticles, then they need to follow the EFSA guidance for nanoparticles. Coming back to NAMs and also the aspect of absorption, what I've said before is very much based on traditional approaches, although we've incorporated in the guidance document room for using NAMs. But in parallel, we also been very active in developing a roadmap for the use of NAMs. It's primarily a roadmap for actions, really to see what EFSA as an organization needs to do to help us move into NAMs, into the next generation risk assessment. So absorption is being discussed in this document, but not too much, more focus on metabolism. So absorption tends to be left out a bit because it's perhaps not as, I would say, advanced, or not as, as we have fewer tools really to do this in an in vitro context. However, we also have a project that's already been launched that, as a result, is coming to an end now. And this is, for example, to use in vitro systems to look at digestion, but also uptake of nanofibers, and also a way to look at the local effects that these nanofibers can have. And the focus of this was on, as you can see, nanocellulose, so it's on nanocellulose as a case study, nanocellulose case study. We've continued this outsourcing of projects, and these are just two projects that have just started now. We're still finalizing all the details, but they will also look at aspects of absorption. It's not the main focus of this project, so other elements such as metabolism, for example, for in the ANME4 NGRA project are highlighted, but there will be some work looking at absorption, looking at models just to study absorption, and the same applies for the NAMs for nano projects. I cannot say more at this stage because these projects have just started. So to summarize, so toxicokinetic knowledge we know is an integral component of the current risk assessment paradigm. What we use it for is really enabled to give us a means of decreasing the number of repeated dose toxicities, that is tests that are being requested to be performed if it can be demonstrated that the absorption is negligible. But it also, if there is absorption, also to allow us also to validate the target organ exposure when assessing in vivo genotoxicity studies. These are of course done when the in vitro ones are not conclusive or give us a positive outcome, a positive response. So really where we are now is that there's no need really for validated or approaches that accepted approaches to work. NAMs can be used to predict absorption and bioavailability of chemicals. We really need those, whether it's simple cellular models or with a more complex organ on chip. And we know there are a lot of advances ongoing there, but we really need to get those up and running, ready to be used in a regulatory context. It will also help us very much to understand and to help us in the future decision making that will be done on NAMs to allow us to do the forward and reverse dosimetry. Really, we need to be able to extrapolate to external exposure, because that's the level where the regulation needs to be done. And then also, let's not forget, then we got microbiota, integral role in metabolism of compound in the gut, and of course that needs to be integrated also in those models, so we can also predict the metabolism of the compound to see if there's no let's say mammalian directed metabolism that needs to be taken into account, involved in the toxicity as well as the absorption. And with this, I stop my presentation, and I thank you for your attention.
Thank you. Thank you, George. Looks like we have, can you hear me?
Yes, very well.
Looks like we have a couple questions. Question one, would you assume a conflict comparing the EFSA exposure approach to the ECHA hazard approach?
Exposure approach, or
yeah, EFSA exposure approach compared to the ECHA hazard approach?
Yes, I mean ECHA. I mean the way ECHA operates is very much in classification and labeling. That's the mandate under REACH. So the approach is more hazard driven than the EFSA's approach, which is more of a risk-based approach. So there's some slight differences. And of course, in risk-based, we need to take much more into account the exposure side. However, I should also say that things are moving very fast on the European level. That we're expecting a new legislation on dealing with the so-called one substance, one assessment, which means that should be a common assessment that needs to be undertaken at the EU level to have come up with a common and harmonized evaluation.
Okay, now here's one I quite don't understand. How would EFSA handle an additive for food packaging being a CLP reprotoxicant 2, but with a NOEL sufficient enough to grant a safe SML? You can translate that for me.
Yeah, thanks for the question. I mean, that's coming back to the issue of one substance, one assessment. We've had in the past some, let's say, some gap. Of course, if there's a concern for genotoxicity, then the substance has no room in a food packaging material. Also, if there's evidence for ED, we've seen that with the BPA situation again, then there's issues, clear issues here identified. But in terms of food contact material, the guidance there is very much exposure driven. Exposure in the sense it's driven by the amount of release from the material, from the plastics. So it's the leaching into the food, into different food categories, and from there the testing is also dependent, or the amount of testing is dictated by the amount of release. So the exposure is extremely low. Then there's the battery is smaller than if it's higher release. But of course, landscape is, as I said, changing quite quickly with the one substance, one assessment. We need to harmonize more the approaches.
So we have one more question, but I'll have you answer. It's from the same person, White Shark. Maybe perhaps all three questions are for him. He should email you and talk about some of these things, if that's okay, because we want to move on to the next seminar. Thank you again, George, for a really stimulating seminar. And I'm sure you'll be hearing from a lot of us on ways we can partner with you. So our next speaker.
Thank you very much, Susie.
Yeah, thank you. Our next speaker is Jose Tarzano. He is research professor and head of risk assessment unit at the National Environmental Health Center in Spain. He's been involved with regulatory assessment since 1992, vice chair of the EU Scientific Committee on Health and Environmental Risks, Chair of the Risk Assessment Committee of ECHA, Head of the Pesticides Unit, and Senior Science Officer covering NAMS of the European Food Safety Authority. He was at EFSA till recently. He is involved now with the EU partnership called PARC. He's the co-chair with me on the work group of NAMS under ILMERAC, which is our international global consortium of food regulators, and a great partner, I might add, for that and other things. So Jose, we're looking forward to hearing from you.
Thank you, Susie, for your very kind introduction. I hope that you can see my slide. So first, I would like to thank Susie and the other organizer of this event for inviting me to give a perspective from the scientific approach of the use of NAMs for mechanistic information, understanding the gastrointestinal tract. As George has already mentioned, most of the regulatory assessments today are done using animal studies. It's the typical paradigm from the 50s, 1950s that was developed using apical endpoints, and the full idea is to try to identify a point of departure of that represent no adverse effect level observed in the animals. On the other hand, toxicology is moving to many other methods, alternatives, what we call NAMs, new approach methodologies, mostly animal-free, but in some cases also combination, for example, of short-term exposure to animals with NAM, with omics. And one of the questions is that these elements need to be integrated into the assessment. It's important to remember that in both cases, those two models, the animal models, but also the human in vitro models, require extrapolation. But the kind of extrapolation that is needed is different. In the case of the animal models, we need to do the extrapolation between the species, so from the animal to different groups of the human populations. And in the case of human in vitro models, we need to do the in vitro to in vivo extrapolation, the conditions in which the chemical are assessed in the in vitro models, obviously, try to reflect the physiological conditions, but are far away much more simplistic and do not always represent the physiological conditions that we have in our bodies. Let's focus specifically on gut toxicity and the animal models. So the main assumption is that the gut processes are covered in the animal studies. Even we know that the microbiota and the physiology of the different species are very different. For example, the capacity for the degradation of fiber or like cellulose in even in rats or mice is clearly much higher than humans. The composition is different, but there are no specific considerations regarding the physiological difference. It has been already mentioned by also Susie in the introduction and yours that a lot of effort is put on the fate, so on the ADME, the toxicokinetic component, specifically the absorption is the more relevant process for the gastrointestinal tract. And for the moment, there is, or in the past, has been quite little attention to the microbiome and effects directly on the microbiota, assuming that is covered in the adverse effects observed in the animals, in the intact animal. Main limitations is for the extrapolation focusing on the toxicokinetics and the absorption, but maybe it's quantitative. We already know that there are some differences, but with much more limited information on the mechanisms for the absorption that can be different as well. A focus on the dose and not necessarily that is the kind of administration is similar. So what we have in most of the studies is either gavage administration or animal in feed, but in any case, the feed that we provide to the experimental animals do not resemble human food. So the combination of the effect of the chemical and the interlink with the food and of the food with the microbiota are not really covered. There are some limitations if you ask particularly to the histopathologists for the real assessment of local effects on the gut, and in most of the cases, lack of information on effects on the microbiome. As George was mentioning, the risk assessment and all regulatory agencies in the world are trying to move to cover the evolution of the paradigm. I try to use the example, basically, if you have an electric car with could be the new paradigm, and you have oil, you cannot do the connection directly, but you can always have a generator, and then you can use oil to produce electricity and move your car. This is the evolution. We keep the standard principle, trying to fix a point of departure, but we include some mechanistic information using NAMs, basically at least that effects observed in vitro or in silico studies are well covered by the in vivo observations. One additional element is that all the studies are assuming that they are conducted on healthy animals and are assuming that the risk assessment is for a healthy population. But that is not the real case. So if you look to the statistics, at least 50% of the adult population, both in North America or in Europe, have at least one chronic disease. And this is not covered by the current paradigm for risk assessment. The evolution of the paradigm means to improve part of the assessment through mechanistic information. The black box, that means that we just administer the chemical to the animal, and then we look to the animal to see what effects we are able to observe, can be open with information of that is provided by new approach methodologies, effects on microbiota, on the gut epithelia, or even the connections in the liver, what's happening also with the circulation of the chemicals from the intestine to the liver, and also back from the liver to the intestine. So the mechanistic information can be incorporated in this traditional risk assessment paradigm. Examples of experimental models, and there are a lot, so I have just put some examples. Experimental models that are used for in these assessments can be divided in three basic groups. The first, the more simplistic, we just isolate some biological component of the system, either the microbiome or the cells, normally the epithelial cells, and we do testing directly, just to expose the organisms to, or the cells to, the chemical. A second step that is moving more and more is to consider, okay, the main role of the gut epithelia is to be a barrier between the lumen and the blood. So let's try to reproduce. And there are different examples. This can be done even in chemical methods, no biological system, just a membrane to see the capacity of the chemical to be moved through it. Or we can construct a real membrane with cells, either a monoculture like a monoculture of cockatoo cells, or a combination of cells that are more closer to the physiological conditions, with cells that are able to produce mucus, other cells that are able to have a microphagocytosis activity, and other cells that can represent different elements. But still, it's just a system, a system divided by a barrier of cells. Or we can try to cover the dynamics as well. We are moving on to the microfluidic system that Susie is a real specialist, or what we call the cat on the chip, or the organoids of intestinal organoids. And then we have a more sophisticated system, in some cases with the capacity to have a dynamic flow, and also even to reproduce peristaltic movement, but nevertheless are simplifications. So the main assumptions when we are using human systems for assessing gut toxicity is that the system reflects the human physiology and is relevant for humans because it uses human models or human cells. But the reality is that the in vitro conditions may be very different from those that are real under physiological conditions. The focus is always on the intestinal epithelia, and there is a little capacity to cover other specific elements like the Peyer patches, or and there is also independent assessment of the effects on the microbiome. The idea is that we use adverse outcome pathways to do the extrapolation from the effects that are intermediate effects that we can observe in vitro to adverse outcomes. And the main focus and the main activity in the area of gastrointestinal tract is for quantitative in vivo in vitro extrapolation, focusing on the toxicokinetics. These systems obviously have also limitations. The main limitation is the simplification. Even the more complex system like the cats on the chip are still far away from the real conditions and the complexity of the human body. Mostly have a quite short time frames. This may be a problem in many of the cases for the extrapolation to adverse outcomes that require some time to be developed. Only the most complex systems you need to have a dynamic situation. But in that case, you can integrate the microbiome in the same system. And finally, provide mechanistic information or quantitative information. But it's normally either one or the other, not a lot of information combining both in the same methodology. There are a lot of efforts to move into this new assessment way and the evolution of the paradigm. The partnership partnership for the assessment of risk, for assessment of risk of chemicals in the EU has been already mentioned. It's a very large multi-activity with a lot of different projects and work packages. Basically, we have a work package 5 covering the hazard assessment and work package 6 covering the risk assessment. In this publication by Philip Stolen, you will have all the details. But if we focus specifically on the work package 5, and also again in this publication from Talia, you have all the details. You see the different sub work packages and the different projects and activities, and you can see that there is a lot of the toxicokinetics and the use of AOPs, but the examples that have been selected are not specifically focused on the intestine. So basically what we can expect from PARC is a lot of advance on toxicokinetics and on a focus on systemic effects, but not specifically on effects on the gut or after the metabolisms and the possibility for considering the effects of metabolites at different levels. There are additional efforts by EFSA that George has been also mentioned. I want to highlight in particular these publications. The last one in particular, because it's a toxicokinetic model that was developed exclusively using non-animal methods. So this is a case study that has already demonstrated that we can already repeat toxicokinetic information without using the animals, and then we have a focus on oral uptake, obviously for EFSA. And EFSA also mentioned that the TKD play, so a model to allow everybody to do these assessments, will be launched very soon. One additional element for the use of these new methods is that the need to have not only very good scientific quality, but also the need to have the methods that can be verified and the results that can be verified by the risk assessors. It's not only the question of the quality, it's the capacity to verify, to replicate the methodology, to have access to the data. And one additional element that I see very relevant produced by EFSA is this poster with the different elements. There are the 7 elements that should be considered by researchers in the area of organ of the chip in order to be sure that the results could be used in the regulatory context. So I think that this information may be also very relevant and useful. And this covered the evolution of the risk assessment paradigm. So how we can try to incorporate NAMs in the current system. On the other hand, obviously what we can expect as researchers is to have a much more development, is what we call the revolution of the paradigm, being able to think out of the box. There is no need to have just a single point of departure. We need to just assess safety, and there are different methodologies and options, and we can simply rediscuss how we do risk assessment and health impact assessments, and try to use all the information that we have today and try to evolve the paradigm into something absolutely new. This is the idea of the next generation risk assessment, that they start with the hypothesis-driven information, goes to the consideration of integrated approaches to testing and assessment to use existing data. Obviously, if we have already animal data, we should use the data, but also we have all the extrapolation with read across that George was mentioned as well. And in this area of new approach methodologies, the question is to use adverse outcome pathways to address the mechanisms. And if we combine all these elements, we may have a different kind of paradigm for risk assessment, in which the mechanistic information is essential to select the drivers for the hazard, and then to move into the risk assessment. Focusing on the gap, there are two elements that should be covered, and there are a lot of research is needed in order to cover those effects. One are the effects on the microbiota, considering the dissolution, degradation, transformation, but also the direct effects and the consequences. And the second is the effects on the gut epithelia, local effects, and in addition to uptake and transfer for the blood. So what the idea of these processes is to identify the information available for the chemical, to read across from similar chemicals, and using non-animal studies, try to identify a problem formulation and drivers for the hazard and exposure that are combined through a new way of doing risk assessments. And as already mentioned, for the hazard part, they all focus on the adverse outcome pathways. But we already discussed at the World Health in episode 2 years ago that one thing is to construct an AOP, and something very different is to use the AOP in risk assessment. The AOPs, for developing the AOPs, are basically a simplification. In most of the cases, I will say that oversimplification, trying to have a single line of events. You see here, for gut, there are not a lot, but this is a very general one for reactive oxygen species and inflammation that produce increased likelihood for cancer. That is also in which one of the elements or the tissues that may be covered by this AOP is gut. And as you can see, it's a linear succession of events. When you move to the risk assessment, what you have is something absolutely different. You will have different experiments on different models, most of the cases at different levels of the biological organization. And the key element for the risk assessment is to identify the effects, but also the effects linked to the particular consideration for exposure in terms of the dose or concentration as well as the time. But then to try to identify the likelihood for the effect to move through the progression of adversity, so through more adverse outcome pathways, so the closer to something that really can be considered as adverse. A compensation with may have additional elements, for example, a compensation of the physiology, but at a particular cost in terms of metabolic or energy consumption or full recovery. And these are the key elements for the different effects that have been observed that need to be considered in the risk assessment, that obviously are much more complex than the simplify AOP or even AOP networks. What are the needs in the area of gut toxicology? Obviously, we need to consider both toxicokinetics and toxicodynamics. For the toxicokinetics, the absorption, we have already several models. There are also models even for in vitro comparative assessment for the metabolisms in liver. The possibility for additional skeleton through the bile and reabsorption is less covered with the current models. And then we need to have a toxicokinetic information for the systemic concentrations. For the effect, we have also some options to assess. In most of the cases, either the effects on the microbiota, the microbiome, or the effects on the epithelia. Only with some cases we need a combination of both, but should be very relevant to try to move into these models that are able to measure both. The possibility also for testing the metabolites generated through the liver or other organs, but even more important to move into the adversity. What are the final consequences of these effects in terms of impacts on human health? And one of the big challenges is to have these complex interactions. You can see here some reviews indicated the link between the microbiome and gut with the axis, with the specific axis to the brain, to the lung, to the cardiovascular system. These are more and more considering as key elements for the impact assessment of on the health at a health level, and the possibility or current possibility to try to replicate this using NAMs is very limited, but also the capacity of animal models to offer information relevant for humans for many of these connections is very limited. So it is one of the elements that we need to clearly progress. So the way that we are suggesting to have this assessment is to consider a different consideration of adversity. It's less important to define if something is adverse or not, but what is the likelihood for this particular effect, this intermediate effect, to move into measurable health indicators, to originate, to move and to progress on something that is really a health impact. And this require a matrix for risk assessment. So this new paradigm, then you have the central is AOP-based adversity indicators. And just to finalize, in order to prepare the workshop, I will suggest some possible considerations. First, we need to consider that the assumptions, limitations, and uncertainties are not only for the use of NAMs. Animal models also have a lot of assumptions, limitations, and uncertainties that should be considered in the comparison. We cannot assume that the animal is the gold standard. We need to consider that both are limitations that require different extrapolation elements. What we have is a significant advance in NAMs-based toxicokinetics, almost ready to be used in the regulatory context, and a relevant number of tools with different level of complexity for covering the toxicodynamics, the effects on microbiota and the effects on the gut epithelia. But what we need is system to qualify those elements that move in the regulatory assessment. And I know that the FDA, EMA, or EFSA are working on that, but also a new paradigm for the next generation risk assessment that should address what are the initial effects, but also what are the consequences associated to those effects when we move to public health. And that's all. Thank you very much for your attention.
Oh, thank you so much, Jose. That was a great talk. And it looks like we have one Q&A. Yeah, same guy. Does your institute have experience with chemicals having an antimicrobial effect, causing malnutrition, triggering secondary toxicity, systemic, and on reproduction? I think this was for both you and George.
I do not have experience on that. So in my institute, I'm not sure if yours have but from my times in EFSA, I don't think that we have this kind of experience. One of the key elements, nevertheless, and this may be relevant, reduction in body weight is one of the most adverse effects that are used for setting the point of departure. So it's one of the more traditional elements that is used as an indication of adversity, but there is no sufficient indication if those is produced to effects of antimicrobial effects or something else. So it's the apical effect is there, is a reduction in body weight or body gain, but what is the mechanism is, in most of the cases, not identified in the animal studies.
Thank you, Jose. I can't really tell if people have hands up, but it looks like we're almost out of time. We are out of time. Thank you again, George and Jose, for really great talks, a really good introduction to our Trust the Gut in-person workshop in hopefully at NIH in October. And we hope to see you guys all there. And remember, both Jose and George's presentations are going to be webcasts, and I'm sure their emails are on there. And I know they'll love to answer anyone else's questions, right? And we'll see you guys soon. Thank you so much. Bye-bye.